What if I told you that inside every single living cell, yours included, there's a hidden library? I'm talking about a collection of historical documents telling a story that goes back billions of years. Now, these aren't written on paper. Nope. They're written in the very machinery of life itself, proteins. So today, we're going to dive into how scientists are actually reading these ancient stories, not from fossils buried in rock, but from the molecules that make you, well, you. So this whole idea really kicked off with this one powerful concept put forward way back in 1965. It was honestly revolutionary. The thought was, what if we could treat the molecules inside of us, things like proteins, as living fossils? What if we could actually read their structure to uncover the entire epic story of evolution? I mean, it's a pretty amazing question, right? How can a tiny, intricate chain of molecules, a single protein, hold the key to understanding the entire vast tree of life? Well, that's exactly what we're going to unpack. Let's get into how scientists actually learn to read this incredible molecular language. Okay, so to read these molecular documents, first things first, you've got to understand the language they're written in. And even more important, you have to understand how that language changes over time. Think of it like learning to spot all the little edits and notes scribbled in the margins of an ancient manuscript. All right, so the fundamental unit of change, the thing scientists are actually tracking, is something called an amino acid substitution. The easiest way to think about it is this. Imagine a protein sequence is a really, really long word. Over millions of years as species evolve, tiny little typos can happen. One amino acid just gets swapped for another. And believe it or not, tracking these tiny typos is the secret to unlocking the whole evolutionary clock. So how do they actually measure this? Well, the core method is surprisingly straightforward. Step 1, you pick a single protein, let's say GROL, that you can find in a whole bunch of different species. Step 2, you line up the sequences side by side and you just count the differences. That's it. You count the number of those little typos. And here's the crucial part, the simple but really powerful logic. The more differences you find, the more evolutionary distance there is between them. And that is the whole idea behind the molecular clock. The theory is that these little substitutions, these typos, happen at a pretty steady rate over vast stretches of time. So, by counting them up, you can basically measure how long it's been since two species split off from a common ancestor. It's almost like winding back time, but you're using molecules as your guide. Okay, so you've done the work. You've counted all the differences between all these species. Now what? What do you do with all that data? Well, you build a map of the history of life itself. Scientists call it an evolutionary tree, or a phylogenetic tree, if you want to get technical. Let's break down how to read one. All right, the first thing you'll notice on one of these trees are the very tips of the branches. These are called external nodes. Each one represents a species that's alive right now, today. We're talking humans, we're talking bacteria, that tree outside your window. They're all at the end of a branch. They're the current end of the line, so to speak. Okay, now follow those branches backward into the tree. See where two lines join up? That spot is called an internal node. The best way to think about it is as a fork in the road, but way back in the distant past. It represents a common ancestor, a species that's now extinct, but gave rise to everything that follows on those new, branching paths. Now here's where it gets really cool, and this is probably the most important part. The links of the branches aren't just random. This isn't just an illustration to look nice. Each branch length is mathematically precise. It's directly proportional to the number of those amino acid substitutions we were talking about. So what does that actually mean when you're looking at it? It's simple. A longer branch means more changes have happened, and more changes mean more evolutionary time has passed. A short branch, that means fewer changes, and a much more recent split from that common ancestor. So you'll often see a little scale bar on these diagrams, usually with a number next to it. At first glance, it might just look like, well, a number, but that little number is the key that unlocks the whole map. It's what gives a precise mathematical meaning to the length of every single branch on the tree. So what does a number like 0.1 actually mean? Well, it means that if you have a branch that's the same length as that scale bar, it represents an average of one amino acid substitution, one typo, for every 10 spots in that protein sequence. This is how scientists calibrate the whole thing, turning what looks like a simple drawing into a really precise map of genetic distance. But, of course, biology is rarely that simple. Here's a question. What happens if you build a tree using one protein, and then you build another tree for the exact same species, but using a different protein?
And, the stories don't quite match up?
Well, the reason for that is that not all proteins are on the same schedule. They don't all collect typos at the same speed. Some proteins evolve really fast, racking up a ton of changes even between close relatives. These are fantastic for mapping out recent branches. But other proteins evolve super slowly. They might be almost identical across huge, diverse groups of life. And those are the ones you use to map the really ancient, deep relationships in the tree of life. And to make things even more interesting, a single protein can actually change its evolutionary speed. It can evolve faster in one group of species than in another. So a tree based on just one protein is really only telling you one version of the story. So how do scientists get around this? Well, they don't rely on just one document. They combine the data from many different proteins, sometimes hundreds of them. This lets them cross-reference all the different stories to build a much more detailed and accurate picture of what really happened.
So at the end of the day, why does all this matter? I mean, why go to all this trouble to peer into the molecular world and trace these ancient connections? Well, it turns out the implications are absolutely profound for how we understand, well, life itself.
And it's so important to remember that this isn't about finding some final, dusty answer in a textbook. The story of evolution is a work in progress, and the science itself is always evolving. Every new genome we sequence, every new protein we analyze, it's like adding another chapter, another layer of incredible detail to the story. And look, that story really matters. This research is fundamental to how we as humans see ourselves and our place in the universe. It shows us in black and white our deep biological connection to every other living thing, from the simplest bacteria to the biggest blue whale. It places us inside this immense, incredible, interconnected web of life. And that's why this field isn't just about looking at the past. It's really about the future of scientific discovery. Think about it. Our technology for sequencing genes and proteins is getting better and faster all the time. Our ability to read these molecular documents is just going to keep getting better. It is without a doubt one of the most vibrant and exciting frontiers in all of science. Which really just leaves us with one final, kind of mind-blowing thought. The history of life isn't just something you find in fossils buried deep in the ground. It's inside you, right now. It's flowing through the veins of every living thing. It's written into your very own proteins. And that just leaves one big question. What other amazing stories are still in there, just waiting to be read?